Here is the first speaker, Professor David Weed from Weed Research Foundation, the United States of America.
My name is David Wade, and my affiliation is the Wade Research Foundation, located in Princeton, New Jersey. The title of my presentation is Constructing a Novel Molecular World Based on Names. The project that I will describe has a scientific basis, and I will begin by providing some scientific background information. In 2003, I invented a method for using the letter sequences of names as the basis for creating physical objects that could be studied by conventional scientific methods. I call it the name-to-peptide method. This slide shows a table that lists the molecular components of an E. coli bacterial cell. Cells of higher organisms, such as humans, have the same types of molecular components and in roughly the same amounts. The most abundant molecule type is water, comprising 70% of the total weight of the cell. The second most abundant molecule type are proteins, comprising 15% of the total weight of the cell. Proteins are polymers composed of amino acid monomers or building blocks. This is illustrated in the middle of the slide by a string of zeros linked together by horizontal lines. The zeros represent amino acids, which I'll abbreviate as AAS, and the horizontal lines between the zeros represent chemical bonds linking the amino acids together in the polymer. Two terms are used to describe the sizes of such amino acid polymers, peptides and proteins. If the polymer contains 2 to 100 amino acids, it is called a peptide. And if it contains more than 100 amino acids, it is called a protein. I will focus on the smaller size polymers called peptides. A well-known peptide is the hormone insulin, which contains 51 amino acids and is used in the treatment of diabetes. 22 different types of gene-encoded amino acids have been found in naturally occurring peptides and proteins. The International Union of Pure and Applied Chemistry, International Union of Biochemistry and Molecular Biology, Joint Commission on Biochemical Nomenclature, abbreviated IUPAC, IUBMB, JCBN, and which I will abbreviate simply as IUPAC, has assigned 4 types of names to these 22 amino acids. The assigned names simplify communications between chemists, biochemists, and molecular biologists throughout the world, and they are used when discussing amino acids, peptides, and proteins. This slide provides an example of the 4 types of names assigned to each amino acid by the IUPAC. The figure at the top of the slide is a chemical structure of one of the 22 amino acids. Its complex systematic name is 2-amino-3-1-H imidazole 4-yle propanoic acid. A less complex and more commonly used trivial name is histidine. It also has been assigned a 3-letter name abbreviation HIS. The simplest designation for the names of the 22 amino acids are 1-letter symbols borrowed from the 26-letter basic Latin alphabet. The 1-letter symbol for this amino acid is H. The name-to-peptide method that I will describe is based upon the IUPAC's 1-letter symbols for the names of amino acids. This slide shows the trivial names for each of the 22 amino acids and their corresponding 1-letter symbols. Notice that all of the 1-letter symbols are uppercase or capital letters. The reason for this symbolism is based on a physical feature of amino acids called enantiomerism, whereby each of the amino acids has 2 possible mirror image forms. Since nature only utilizes one of the 2 forms to construct peptides and proteins, the IUPAC nomenclature symbolizes this form by uppercase or capital letters. Since there are only 22 different types of gene-encoded amino acids, and the basic Latin alphabet contains 26 letters, the letters B, J, X, and Z do not have unambiguous assignments in the IUPAC nomenclature. B can represent either of two amino acids. J has not been assigned to an amino acid. X can represent any amino acid, and Z, like B, can represent two amino acids. Since these four letters have ambiguous representations or have not been assigned, they are not used in the name-to-peptide method. What is the effect of excluding these four letters from the name-to-peptide method? Using the one-letter symbols for the names of amino acids, this slide compares the frequency of occurrence of amino acids in vertebrate proteins, shown as dark gray bars, with the frequency of occurrence of each letter in English language text, shown as light gray bars. I was not able to find any list of the frequencies of occurrence of these letters in names. The graph shows a similar low frequency of occurrence of B, J, X, and Z, highlighted by red squares, among amino acids and among English language text. Consequently, the absence of B, J, X, and Z from the name-to-peptide method is not expected to significantly reduce its utility. The average size of proteins varies from 283 to 472 amino acids, and the most efficient method of storing the amino acid sequences of these proteins is to use the one-letter symbols. The U.S. National Center for Biotechnology Information's computerized protein database contains the amino acid sequences of 875,680,339 proteins as of February 17th, and all sequences are stored as strings of one-letter symbols. I will now use one example to describe the name-to-peptide method and its ability to generate novel and potentially useful physical objects based upon the letter sequences of names. The first step of the method is to consider any string of letters as a potential sequence of amino acids in a peptide or protein. For example, the name Walmart is the name of an American multinational retail corporation and the world's largest company by revenue. The company name consists of 7 letters, each of which is also a one-letter symbol for the name of an amino acid. The corresponding trivial names of those amino acids is shown in the middle of this slide, and the respective one-letter symbols are shown in parentheses next to the trivial name. This slide shows one type of chemical structure for peptide Walmart. The one-letter symbol for each amino acid is shown in red next to the corresponding amino acid in the peptide. This slide shows some predicted physical properties of peptide Walmart, but once an amino acid sequence of a peptide is known, it is possible to predict numerous physical, chemical, and biological properties for the peptide. Does peptide Walmart occur in nature? A search of the protein database did not find the peptide by itself, but did find it among the amino acid sequences of 1,155 proteins. Therefore, the sequence of amino acids represented by the letters in Walmart is compatible with nature. 68% of the occurrences of peptide Walmart in the protein database are within proteins of the bacterium Pseudomonas aeruginosa. This bacterium is medically very important and one of the bacteria that has become resistant to commonly used antibacterial agents. It is a major problem for patients with cystic fibrosis. For researchers involved in the development of new antibiotics, it is very helpful to know the three-dimensional or 3D structure of proteins that might be used as targets for new drug development. One of the proteins from Pseudomonas aeruginosa that contains peptide Walmart has a known 3D structure. This slide shows a ribbon diagram of the actual 3D structure of one of the proteins of Pseudomonas aeruginosa that contains peptide Walmart. The structure was published just last month. The portion of the ribbon that is colored blue represents the Walmart amino acid sequence. This protein is an enzyme or biological catalyst that is essential for the survival of the bacterium. The bottom of this slide shows the biochemical reaction catalyzed by the general category of enzymes shown in the previous slide. However, there is not enough information yet known about the protein shown in the previous slide to understand how the peptide Walmart segment might participate in the reaction. This slide shows the predicted 3D structure of peptide Walmart that was published in 2015, and it is overlaid with the structure of the Walmart amino acid sequence from the three-dimensional structure of the enzyme shown in the previous slide, which was just published 1 month ago. The structures are essentially the same, which illustrates the power of the predictive algorithms used in modern computational chemistry. In 2015, peptide Walmart was chemically synthesized and shown to have anti-cancer activity against human female colorectal adenocarcinoma cells and human male and female urinary bladder carcinoma cells. It was also shown to have potential antimicrobial activity against the bacterium Staphylococcus simulans. This bacterium is an opportunistic pathogen among animals and occasional source of human urinary tract infections. In 2020, during the COVID pandemic, I wondered if peptide Walmart might not be a potential antiviral agent. I did an in silico or computational chemistry experiment that used a program called ClusPro2 running on a supercomputer at Boston University to determine if peptide Walmart might bind to a protein on the surface of the SARS-CoV-2 virus called the spike protein. This viral protein is thought to initiate an infection by binding to a human protein called ACE2 that occurs in many tissues of the human body. This slide shows a cartoon of the SARS-CoV-2 virus on the left. The turquoise colored structures on its surface are spike proteins. The three-dimensional structure of a spike protein is shown in the middle of the slide as a red colored ribbon structure. An infection is thought to begin when the spike protein binds to human ACE2, shown as a blue colored 3D ribbon structure on the right. Certain crucial amino acids are involved in the interfacial interaction of the two proteins. The ClusPro2 program generated 55 binding models, 7 of which had peptide Walmart binding to crucial amino acids on the viral spike protein that are involved in its binding to the human ACE2 protein. This slide shows those 7 models. The viral spike protein is shown as a blue ribbon, and the Walmart peptide is shown as a much smaller red ribbon. The binding of peptide Walmart to the crucial amino acids on the virus spike protein might block the first step in a COVID infection. Further research is needed. This slide summarizes the theoretical and experimental work done with name-peptide Walmart during the past 10 years. In 2015, theoretical analysis of candidate name peptide Walmart were done. It was found to occur among the proteins of a large protein database, and its 3D structure was predicted. The predicted structure was verified this year. In 2020, theoretical analysis of peptide Walmart as an antiviral agent was done, and it was found to be a potential antiviral agent. The peptide was chemically synthesized in 2015, tested in several laboratories, and found to have anti-cancer and potential antimicrobial activities. This slide summarizes the name-to-peptide method for any candidate name. A name is first examined for the presence of the letters B, J, X, and Z. If any of these letters are present, the name is excluded from consideration. If none of these letters are present, then all non-letter spaces and symbols are removed and the letters in the sequence are capitalized. The result is a name peptide that can then be subjected to theoretical analyses synthesized by chemical or biological means, and laboratory tested. Other name peptides have been studied, created by synthetic chemistry and laboratory tested, and some information has been published about them. For example, a name peptide Henryk, spelled Henryk, was synthesized, found to have metal binding properties, and published by a research group in Poland in 2016. The results obtained by considering the letter sequences of just two names, Walmart and Henryk, as amino acid sequences of peptides demonstrate the enormous potential usefulness of the name-to-peptide methodology and hopefully represent an entirely new branch of onomastics.
Okay, thank you very much. Thank you very much. This is very in-depth introduction to a new interdisciplinary research about onomastics and the biochemistry. I'm not quite sure because it's out of my scope.
Yes.
I just have one question for you first, and then I think the audience will have many more. Okay. I still wondering why you assign A to Elaine and the K to asparagine or something like that. You signed different letters to different chemicals, right?
Yes.
But why? Yeah. Can you specify a little bit? Thank you very much.
Yeah, in some cases, when you have the three-letter abbreviation for the trivial name, you can use the three letters that occur at the beginning of the name. But in some cases, it's not used. And these IUPAC nomenclature was developed a long time ago, and for some of these, the ones that do not correspond to the trivial name letters, I just don't know the origin of that. It's not for any particular very important reason. It's just a matter of convenience, I think. So I really can't answer that particular question.
I have a question in the chat box. It's from Cleveland. Is there any advantage to using names for the over non-name words or random generated strings of letters? Thank you.
Well, yeah, that's a common question, and I'm asked if you just use a random selection of letters, would that have any properties? Yes, it would. Because peptides or proteins are so common in biological systems, essentially they are the machinery that drives all the chemistry that occurs continuously in living systems. Every single peptide will have some kind of biological activity. It's just a matter of finding it. You have to do the appropriate test to find it. So the reason there are a whole bunch of reasons why I have chosen this particular area, the use of names. I think once a student finds out about this nomenclature, and there's probably about 500,000 people just in the United States alone who have been exposed to this nomenclature, when they begin to look at it and begin to look at strings of amino acids in protein databases that are represented by strings of letters, for the first thing they want to do is to see if they can find a name or a personal name or company name, something like that. So that was my original idea, and then for various reasons, a couple of which is the fact that I don't have my own personal laboratory, and because peptide research is extremely expensive, much more expensive than DNA or RNA research. It's difficult unless you are independently wealthy to make these peptides yourself. So it's just you can do a lot of computational chemistry or theoretical analysis with an amino acid sequence before you actually do something in a laboratory with the physical peptide. And in fact, this is what pharmaceutical companies do on a routine basis. Why spend a lot of money doing something if you can test it ahead of time on the computer to see whether it's going to have the activity you're looking for? So I just stuck with that. And whenever I have sufficient funds, I'm able to actually purchase peptides from companies that make them. So far, I've had about 4 that have been synthesized for me. I have published information about 2 of those. 3 of those I have data that has not been published yet. So basically, I stick with it's an easy way that you can explain to students this whole nomenclature, this IUPAC nomenclature, at least for amino acids. If they begin looking at names, something they're familiar with, and then apply that to the nomenclature observation of amino acid sequences in proteins and peptides.
Thank you very much. Personally, I believe that you're opening a new area, okay? It's a really innovative adventure into 2 totally different disciplines. And the scholars will follow your steps and have more research, okay?
Thank you. I think so. Best of my knowledge, I'm the only one that has published anything about it in the US, and there's one other group in Poland who has done the similar thing after I started it. But if peptide research was less expensive, there would be a lot more people involved.
Yes. And I hope that it will inspire scholars in different fields to have such an innovative research method. And, that's really inspiring. And
Thank you.
yes, very inspiring.
Thank you.
Thank you. Or we can wait.
I think that would be just fine. I do have another question for you, David. You mentioned there's a group in Poland that's doing this work. Are they using Polish words and doing the same sort of translating through Polish words, or do they use English words?
Actually, I haven't spoken to them since shortly after I discovered the paper, which was published in 2016. I did ask them if they would like to collaborate later on. They said yes, but I haven't spoken to them since. So they used just a common spelling of a name, Henryk, Henrik. Turned out that was the given name of the head of the laboratory that they worked in. So, personal gratification. But I don't know if they have done anything since. If they they have not published it. One of the problems with doing this particular kind of research, if it's name oriented, is the fact that people in science or peptide science that that's their daily work, do not consider this as something as a serious science, but it is. In a sense, it's completely valid. I do everything that every other scientist does. It's just that the origin of the whatever you're studying, a physical object that you're studying, is a sequence of letters in a name or a word or anything like that. Routinely, these peptides are obtained from nature. They're isolated. Their amino acid sequences are determined, and then they are studied. Here, we're starting not with nature. We're starting with text.
That's really interesting to hear because, I mean, coming from the field of religious studies, you just sort of speaking in general terms, I mean, I can think of a few examples off the top of my head, but in general terms, there are some religious groups that imbue words and names with significant power.
Yes.
I think of even the followers of Rebbe Nachman of Breslov. Put a lot of sort of energy and strength into this one sort of phrase. It's sort of a rendering of his name. If you drive around the modern state of Israel, you'll see it graffitied everywhere by this group that goes around and spreads joy, and that's their sort of one goal in life is to their greatest mitzvah is to just be joyful. And I sort of wonder with names such as that, that have such a sort of a mystical kind of power to people, to people of certain religious groups. If that would be sort of worth looking into or trying out through this name to peptide translation.
Yeah, interestingly that you mentioned that there is the two Hebrew spellings for the name God. If you use the basic Latin alphabet spellings, I did a theoretical study on that, and that of all the little papers that I published, that has the highest number of readings. So for some reason that's attracted attention. I haven't actually synthesized that, but that's an idea. I might try it.
Thank you so much for humoring the suggestion, because yeah, I can just think of, and I can think of other examples too from other traditions that perhaps sometime down the line we could talk about.
Sure.
Cool.
Yeah. Thank you very much, Brendan. Thank you very much, David. Thank you very much for all the participants. Your questions are very important for our session. Time is very limited. I hope that more researchers will go on with your steps. Okay.